To have Professor Emilia Entcheva, who came to speak to us and visit with us from Washington, D.C., George Washington University, always Washington. And I've known Emilia for quite some time, and she's originally from Bulgaria, received her BS and MS in electrical engineering at the University of Sofia. Right. And then came to this country, went to the University of Memphis, where she got her Ph.D., followed by a postdoc in Leslie Klein's lab at Johns Hopkins for several years. And then after that, Stony Brook, she there developed her laboratory for optogenetics and optical imaging, was the director of this laboratory and a full professor, and more recently moved to George Washington University, pretty much developing similar techniques and similar research. So, thanks for coming.
Thank you. It's a pleasure to be here. And I have enjoyed all the meetings leading up to this. So today I'll talk to you about massively parallel all-optical cardiac electrophysiology. So from an engineering point of view, in order to fully understand the biological process, we ideally want to have means to be able to image it or sense it, and means to also be able to perturb it, what we call an actuator of that process, and ideally to be able to close the feedback in order to fully control it. In the field of cardiac electrophysiology, the implementation of that actuation and sensing into a full feedback controller was done through the patch clamp, and that has been the voltage clamp, patch clamp, has been the technology that has allowed us to understand how single cardiomyocytes work. However, cardiac arrhythmias are much more complex phenomena over space and time, and it's not always trivial to extrapolate from single-cell measurements how things will look like at the level of the tissue and the whole heart. So the long-term goal for my group is to be able to do high-throughput cardiac electrophysiology in the multicellular tissue level setting or context, ideally without isolating the cells. So here is a single myocytes, and that's where the electrophysiology field has built its scientific basis and has derived all the relationships. Ideally, we want to be able to study the single cells within their context because we think that is important. And we are looking for tools that are able to do that, to be able to perturb and sense from this multicellular preparation simultaneously from thousands or millions of cells simultaneously. Why is it important to do high throughput? So single cell electrophysiology is notoriously low throughput. Manual, a very experienced patch clamper would do, I don't know, 8-10 cells a day. And when you think about it in statistical terms, that is not meeting any criteria for reproducibility or scientific rigor. One might wonder how have we built all this out of such select information. So NIH has put a lot of efforts to emphasize things like reproducibility and scientific rigor, and the tools that we are trying to develop aim to address these things, this particular two criteria. And here is Christina, who is leading an effort in the lab to be able to do this massively parallel screening of cardiomyocytes that are completely blinded, that we are completely blinded to when we throw in drugs at them or different doses, and try to develop relationships out of that. So the way we approach the problem, how to do this high-throughput electrophysiology, we sort of narrowed it down that optical techniques might be able to give us the means to both perturb and record from with high throughput. The idea of using optical sensors and actuators has been around for quite a while. This particular image is from Gero Miesenbock's paper, where he envisions building a controller around this. And it doesn't have to be membrane potential, but in the context of electrophysiology, that's what the ultimate controller is, because electrophysiology captures this very fast biological processes, which are a challenge to build a feedback controller for. The tools for optical actuation and sensing have been emerging during different decades, I would say. But a critical development happened around 2002 or 2003 when a class of microbial light-sensitive proteins, or opsins, were discovered and cloned that allow the conversion of an energy of a photon into ion flux driver, and therefore control of the electrical properties of the membrane. So in other words, gave us the means for doing the optical actuation. And that, combined with optical sensing, opens the door for what we call potentially all-optical cardiac electrophysiology. Because both techniques are optical, they permit very high spatial temporal resolution of perturbing the tissue with single cell resolution if we wanted to, and sensing what is happening, and then potentially going into wave control at that point, having these tools at hand. Over the last 7 or so years, my lab has developed a number of techniques, both experimental and computational, and these are the people that have been involved in this project over the years. Example here is we developed a computational model of channel rhodopsin 2, one of the main opsins, this optical actuator tool that is able to perturb membrane potential in cells. And we put that model in one of the models developed by Dr. Rudy's group. And here we show what this guinea pig ventricular model is supposed to do when we shine a pulse of light. It triggers this action potential, and this is how channel rhodopsin looks like during the application of that light pulse. And then we measured experimentally, and we show validation of our model within a cardiomyocyte. So this is experimental triggering an action potential, and then we applied what we call optical action potential clamp. That is not only driving a particular recorded action potential, but reproducing the light conditions as well on top of that, in order to extract how channel rhodopsin looks like during the triggering of an action potential. We have gone on to, in collaboration, with Natalia Traianova's group at Hopkins, to integrate this in a whole heart model and be able to show how light can drive the heart. So for today's talk, I will talk about 2 specific projects. We have a number of other things. Oops. This is a little counterintuitive, the buttons forward and backward, but we have a number of other projects ongoing that I won't have time to talk about, but these are the 2 topics I would like to cover. And since some of you were at the Gordon conference a couple of months ago, and I went in this particular order where I ran out of time for this one, so I decided to swap the order for today just for the benefit of those that might have seen part of this talk. So This particular first application is collaboration with Dr. Gilbert, who used to be at Oxford at the time when we started collaborating. He's now at McGill, and we continue collaboration. The idea is illustration. What can one do with these all-optical tools in order to achieve a fundamentally different type of arrhythmia control that is currently not available with the tools that we currently have? And we call this wave steering by light, and I will illustrate what that means. So the current thinking about arrhythmia control, be it pacing, induction of arrhythmia, or termination of arrhythmia, any of this, resynchronization therapy, cardioversion, or defibrillation. One thing is common, that we think about arrhythmia control in terms of static patterns, static over space. So we apply stimulation at location X, and we pace or defibrillate or whatever. So that is how we think about it. The pacemaker is at this location, and we don't move the stimulation. Interestingly enough, even in computer modeling, normally you stimulate from a site or you create a field. You don't move things around in terms of stimulation. So I will show how the light application can change this. Before that, here is an interesting illustration of the spatial temporal resolution that you can achieve by optical means that you couldn't have done by other means. So the idea is that you can project any pattern onto the tissue. It could be truly any pattern. This particular pattern is called Siemens star, and in optics is used to probe for resolution. If I draw concentric circles here, the innermost is telling me how contrast is preserved between the black and white. So in this example, we projected checkerboard pattern onto cardiac tissue, where only the white squares are excited and the black squares are not. So I'm stimulating through the white squares only. The pre-pulse pattern was some fibrillatory pattern in a monolayer of cardiomyocytes. So these colorful things are little wavelets where the black is the wavefront and it's phase mapping, so this is the back of the waves. It's multiple wavelets, slow conduction, and we are applying just the checkerboard pattern. So I'm clamping and defibrillating, if you wish. I'm terminating the waves because I'm clamping the potential at the white squares. So I'm arresting all activity within the white squares, but because of the super short wavelength of this particular arrhythmia, I'm pushing outwards the little wavelets, and this particular arrhythmia continues going on outside within the black area, because I have given it enough space to keep existing. So you can envision what kind of questions can be probed with this technique that absolutely would not have been possible by the traditional electrical or chemical means. I can start growing the squares or start playing with patterns that are able to capture arrhythmias. Right. So moving step forward, it's the key that you can do with light. You can start thinking about the application of the stimulus instead of a static thing, the stimulator to be at this location. You can start thinking of it as a movie. So you can start thinking of applying a movie onto the tissue, a movie of stimulation onto the tissue. And that is fundamentally a new concept if you think about control of arrhythmias. So instead of thinking how can I perturb the action potential of an individual cell, now I'm thinking how can I control the wave, which is a macroscopic property of the tissue, by this moving wave. So the paper that we published with Gil is utilizing a particular setup where our sample is here. We are imaging the sample through a camera, and we use a digital micromirror device, a light projector, just like in this projector in this room, to project movies onto the sample. And the observation from the camera is fed onto a computer where we are seeing what kind of arrhythmia do we have ongoing and what kind of a light pattern do we want to project. So there are details here that this was done by imaging with dye-free technique instead of voltage-sensitive imaging that I don't have time going into. So using this setup, we demonstrated, we predicted computationally here what can you do with this, and then demonstrated in this paper that we can actually manipulate things like conduction velocity by light. Again, we are not just changing the membrane potential thinking in terms of single cells or locations, but we are altering the properties of the waves, the emerging properties of the tissue. So conduction velocity, modulation by light, shown here is increase in conduction velocity with light levels. We can alter the direction of the waves, so we can reliably create conduction blocks. At this time point, we want a conduction block right here. We can create it. Then we want it right here. So we can play different scenarios in this. And the sort of the marketing example is we can change the chirality of a rotor. We can take a spiral wave and flip it back and forth, which is the ultimate control that sort of can be illustrated here. So this is a movie that Gil recorded, and I will play it and I will explain. So the ongoing wave is a spiral wave that we're seeing in a monolayer of cells. And when you see a bright thing coming, is that projected light movie through the projector, which takes the wave and flips it, flips the chirality, and then we do that back and forth. This is the moment of light application, then the wave keeps going, flip it back, and flip it back, and then again. So these are activation maps as the wave was rotating in this direction, in this direction. We applied counter wave by light and let go, and we have flipped the wave effectively, and then back and forth. It's not an example that you go to a clinician and say, this is very useful for the clinic, but it's an example illustrating the level of control that you can have, because you can take that wave and actually navigate it to an obstacle and kill it without defibrillating in the traditional sense. So therefore, we call this wave steering, because it's fundamentally different from what currently can be applied in the clinic. So the point of this part of the talk is that you can design very powerful biological simulators in an experimental system. It's something, in a way, it can outperform computational models in terms of easiness of implementation and speed. Yes, it will depend on the experimental system that you use, but you can play out many, many different scenarios, what to do and what would work on a particular arrhythmia or not. So I envision the use of this technology as a live biological simulator to be widespreading, I'm hoping, and to be useful for validation of computational models in the future. In order to take this from the monolayers to a three-dimensional system, we have developed adult rat model where we make the heart light sensitive. And the transduction is across the, here the lights are bright, but from the epicardium to the endocardium and across the heart. And in an open chest configuration, we can go access with the light and be able to drive the activity onto the heart. So this is ongoing project to be able to do in the live, to do this in the live heart. There are challenges if you want to scale this up to larger hearts. Optical tools have limited depth of penetration. So we are running some simulations. Here is a Monte Carlo simulation based on the properties of the rat heart. This is the absorption spectrum for rat heart and the scattering spectrum within the visible range. Hemoglobin is responsible for this particular peak here. So the optical actuators and optical sensors that we want ideally have to be redshifted, but not in this 550-ish peak where the blood really dominates the effects. So we are working on probes that can be redshifted, including in collaboration with Dr. Corbo at WashU. In collaboration with Pat Boyle and Natalia Trayanova, these are different scenarios. What would it be the benefit of having this optogenetic means of perturbing or stimulating the heart? One benefit is the specificity. For example, if you envision that we specifically transform the Purkinje system to be light sensitive, there is a configuration where if we target electrically the bundle of His here, we can get a substantial energetic benefit in driving the heart by light, because we can selectively target only the Purkinje cells and none of the ventricular myocytes. So computational work in 3D helps us look where to take this technology further. The talk up to here involves exciting things that you can use for mechanistic type of questions, but it's not, in my mind, easily sellable for use in patients because it involves gene therapy. We have to make the patient's heart light sensitive. There are many hurdles to that. From the very beginning, I saw the translational potential of this all-optical electrophysiology in terms of the high throughput that it offers. And that the application of that is for drug discovery, cardiotoxicity screening, characterization of patient-derived, iPS-derived cardiomyocytes, and improving their maturity. So the second application is the translational application indeed. So my student Alex Klimas was really the instrumental one in developing this platform which we call OptoDyCE, which stands for All-Optical Dynamic Cardiac Electrophysiology, which is a fully automated system to go through thousands and millions of cells, be able to optically pace them and optically record variables of interest from them. So we see the big advantages of staying all-optical in the scalability and throughput of such a system. A slide of why do we think there is market for this. You might have seen this number. To take a single drug from preclinical stage to market approval takes on average 12 years. It costs for a single drug in the US $2.9 billion. That percentage of the screened compounds ever make it to market. Only 30% of the drugs that make it to market actually recoup the R&D cost, and so on. So it's very costly, it's very inefficient process, that needs to be optimized in some way. One way would be to remove regulations, as some people higher up think. Other ways is to improve the preclinical stage of screening. A promising detail here is that if you involve what they call biomarkers, in other words, if you make the experimental model in any way relevant to the patients that you're designing drugs for, be it by gender, by race, by other biomarkers, you instantly increase the probability of success for that drug. So here is the depiction of the current pipeline to develop drugs. You start with some compounds. There is some screening here that, if done manually, is extremely low throughput. Is usually in heterologous system, in hex cells, where a single channel is introduced, ERK channel for cardiotoxicity, and it's very costly. There is attempt to automate this, to scale this up, but it's not meeting the criteria for truly high throughput. Then there is the preclinical animal testing, which I guess my students took extra care to select very cute rat and dog. And then it's the preclinical human testing, which is always done in male, young, healthy males, the safety testing, and then we go to clinical trials. Here are the price tags for each of the stages, this one, of course, being the most pricey. So the idea that we have is where our system comes into the picture and how biomarkers are being brought into the picture is to leverage the developments that have been ongoing. This is work in progress, to derive patient-specific iPS-derived cardiomyocytes and do high-throughput screening on those. This is our estimate for such screening and the benefits of doing things this way. Ideally, once validated, this part should be able to sort of replace this plus this before going into clinical trials, ideally. So the system, the OptoDyCE system, looks something like this. So here is a schematic of our system. So we make cardiomyocytes light sensitive by our means so that we can optically paste them and optically record from them. And then the efficiency is so high that the actual system is extremely simple. We use LEDs only, no lasers. This one is the actuating LED. So we have spectrally combined multiple things. We have been able to pack them together. Blue light for pacing, green light to stimulate and record calcium, red light to stimulate, and we have a near infrared dye to record voltage. All that is done simultaneously onto a single detector. So this is the pacing LED, calcium monitoring, voltage monitoring LED, some optics, single detector. We are doing strobing, so we are recording onto the same detector the voltage and calcium. And these are some records from human iPS-derived cardiomyocytes in monolayers. We are optically pacing them. These are the voltage transients, and these are the calcium transients from these cells in monolayers. The key point is that we automated the system, so it's a push-button type of system at this point. And you can put a 96 well plate or 384 well plate or whatever format you have, or a single dish, and it will go through the plate without you having to do things. So it's enormous amount of information that could be collected in, let's say, 10 minutes for a 96 well plate. when we do the calculation, how many cells can we do per day, we actually theoretically can meet the criterion for high throughput. We can test 10,000 compounds a day. It's not something we are actually doing. We do not have robotics. The student is depositing cells manually, so it's Alex is the robot right now. But the potential is there. The technology definitely allows for high throughput screening for the first time. So such thing is not available by commercial means. The parameters of interest that one can measure with this system include voltage, calcium, and contractility. These are some examples from our paper, actually. This was done with neonatal rat myocytes, where you can capture some abnormal events in them, in the voltage. You can capture calcium alternance in this human iPS-derived cardiomyocytes. And here are abnormal contractions that you can capture in these cells, all measured by optical means. Because of the dynamic aspect, we can drive the system to instabilities. So it's very important when you do arrhythmia testing or drug testing, the element of being able to dynamically pace these cells to instabilities is extremely important. So the high content information reflects pacing rates, multiple parameters being measured, multiple doses of a drug being measured. Example here is nifedipine, 12 different doses. As I said, this plate would be read in 10 minutes. And you get instantaneously what does it do on the voltage, on the calcium. And you can construct curves, push it beyond the physiological limit. For example, for nifedipine, this was sort of interesting for me to see that beyond a point, beyond 1 micromolar, you actually start recovering the APD back, perhaps due to saturated action on the L-type calcium channel block, and you start having non-specific effects or the effects of nifedipine that are not meant for the L-type calcium channel. So in attempt to make this more robust, we have a way to not only transform the cardiomyocytes to sense the optical pacing, but what we call spark cells, so we can have a non-myocytes that are dedicated light-sensitive cells, which when sprinkled for a short period of time with cardiomyocytes, can give us the means to do the same. So here is side-by-side comparison for nifedipine dufetalide with rat cells. When on the left column here, we are doing this with virally transduced cardiomyocytes, and here we are doing it through the spark cells that allow us to drive the cardiomyocytes without the cardiomyocytes being genetically transformed with the light-sensitive channels. And we validate that you get qualitatively, in many cases quantitatively, the same response in voltage and calcium. So it's just another way that might be better in terms of industrial application of this. One has to worry if we are genetically modifying things and we are doing drug testing. Many of these opsins are actually derivatives of ion channels themselves, of microbial ion channels. So you might ask the question, what if the drug unknown drug that you are testing is not acting on the optogenetic tools themselves rather than the myocytes that I'm interested in? So we have undergone some validation to prove that this optogenetic manipulation is benign. Here was computational side-by-side comparison that electrical stimulation, optical stimulation above threshold are identical. And then experimentally, when we optically pace and electrically pace, the action potential and calcium transients look very similar. Then we did sort of computational experiment to, again, validate this. I imagined a drug that is very severely changing the kinetics of channel rhodopsin, of diopsin. Severely slowing down kinetics, distorting it, and then we look what does it do to the action potential and the calcium transient of the cardiomyocyte. And the effect is completely minimal. The reason for that is because of this general time separation. If I'm to pace these cells by light, things that happen afterwards are very minimally affected by what the channel rhodopsin is doing. There is some residual effect if I slow down the kinetics of channel rhodopsin extremely, but the effects are minimal. On the other hand, if I'm using an optogenetic sensor, which in some cases we do, we have our gecko to record calcium from these cells simultaneously with the optogenetic pacing, they're completely time convolved. So the optogenetic sensor is co-present with the response of the myocyte, and therefore if the drug, this is just fictitious simulation, if the drug is doing something to this, we implemented this particular voltage sensor, VSFP, computationally, it's severely distorting the action potential because of this time convolution. So one has to be aware what is allowable in this drug testing domain, but by all means, it seems that the optical pacing is perfectly fine. So the platform is experimentally, impartial to the experimental model used. We decided to go with human iPS-derived myocytes because of multiple reasons of relevance and scalability, but the same platform can be applied to, let's say, cells or tissues derived from donor human hearts. We certainly have applied it to dog-derived cardiomyocytes, to guinea pig cells, mouse cells, and rat cells. So because it's an optical platform, it can work in 3D. There is no contact, so we have grown cells in small 3D constructs. As long as it's within 500 microns, we can work with it. So these are different experimental models that are compatible with the technology. So in order to use this towards something of relevance, some of you might be aware, the FDA is working towards revamping the way cardiotoxicity testing is done. And the main concept is that so far the ERK channel testing has been the required one. So one channel, the main culprit usually in cardiotoxicity, is being tested. So they want to run multiple ion channels, then use a computational model such as the O'Hara model, for example, to integrate the single channel information in order to make a prediction at the cell level, is this drug safe or not. They have a second arm from the right here to use human iPS-derived cardiomyocytes to do such a prediction and compare both arms and decide where to go. There are many unknowns during this integration part. Those of you that develop computer models know that there are many unknowns. There is a lot of signaling that we have not captured so far within the computer model. There are several processes that are not necessarily reflected how the cell operates. So it is attractive to go from the right with the iPS-derived cardiomyocytes, but currently there are no high-throughput capabilities to do that, and that's where we see our platform fitting. So this last month, between the Gordon conference and now, Alex actually, we signed up to be a testing site for this SIPA initiative, and she used 3 days with no robotics to test blindly 12 drugs, 5 doses each. When you take into account the number of samples and controls, it's roughly 500 multicellular samples, not 500 cells, that she did. And this some of, we don't know what this compound is. At the highest dose, this particular compound caused this kind of arrhythmias. This is in human iPS-derived cardiomyocytes, in DMSO, the low-dose positive control dufetalide, and we are recording calcium in red and voltage in black. Again, this is from, this is actual experimental signal, not simulations. So voltage and calcium from the human iPS-derived cardiomyocytes. She has tons of data to analyze. I do not have the results from that yet. So one example that I'm running late. So one example that I wanted to show is a case study of a drug that went all the way to phase 3 trial, and these were things that were tried before it was deemed safe to be put in patients. The drug is vanoxerine, and was supposed to be illustrating the concept that FDA has, instead of doing ERK channel testing only, to expand the testing to multiple channels. So vanoxerine is known potent ERK channel blocker, but it's also a blocker of calcium and sodium. And the idea is, if it blocks both forces, perhaps it's actually safe. And has been shown to be safe through these different steps, preclinical steps. It failed end of 2015 in phase 3, where several patients developed severe TDPs. So we wanted to throw vanoxerine, and the idea was, could we have caught this? So vanoxerine in our hands prolonged the action potential substantially, reduced calcium transients substantially, and then we looked closely. It seemed to lead to some sort of uncoupling between the calcium and the voltage, where in the voltage you see this EAD type of bumps that are not necessarily seen easily in the calcium, or the other way around, this bumps into the calcium. So this is at the single cell level within the multicellular preparation that we are observing. And in many cases, it was doing really crazy things, especially in the calcium. So because of the ability to observe the behavior of individual cells within the multicellular preparation, we looked how synchronized things are. So we were interested if a drug alters the coherence across space. And so we look in terms of spatial coherence of the action of the drug. So color-coded here is, so high synchrony is blue. So under control conditions, everything is coherent. The sample behaves the same. If we pace it, things are simultaneously going at one frequency. When we start adding vanoxerine, there is increased incoherence, so reduction of spatial coherence, or this heterogeneous appearance, especially in the voltage domain. And that is very reminiscent to how visually the positive control dufetalide looks like. So this is a map of the loss of spatial coherence with dufetalide application, or Bepredil, another known pro-arrhythmic drug. So this is one view where a multicellular preparation can have a signature of pro-arrhythmic effect of a drug that we are exploring. So different ways of analyzing this data. And this particular dose of vanoxerine was well within the limits of what was actually administered to the patients. Because we chose to use iPS-derived cardiomyocytes as our preferred experimental system, the question always comes up in any cardiac audience, but they're immature. So that is the criticism, legitimately so, because they have a lot of issues. So the question is, can we use our technology to help address this problem? And there are groups that are pursuing any of this or combination of this in terms of increasing maturity of this iPS-derived cardiomyocytes. This is a big one. It really matters what is in the media, and the companies that sell these cells have proprietary cocktails usually. Perfusion, growing them in a particular way, innervation, changing the extracellular matrix, growing them with fibroblasts, 2D versus 3D, loading them, all of those. So all this is complicated. So we like to go simple about this, and we ask the simple question, how about cell density? Does it matter whether we study isolated cells versus cells within the tissue context in terms of their maturity? So we plated cells at different cell density and used this high-throughput system to go through many samples to uncover, does the cell density matter in terms of the spontaneous frequency of these cells? So often these cells beat on their own. These commercially available cells that in our hands beat very slowly, so they're consistent with what a ventricular cell would do if extracted and doesn't have a driver. So it's relatively low frequency beating. But the moment we increase the plating density, the frequency of beating definitely was affected. So they beat less. Things that are affecting this spontaneous activity as a signature of immaturity of the cells that have been proposed in the literature. A big one is the claim is that these cells do not have IK1, and therefore it's very easy for them to beat. Early simulations by Dr. Rudy and John have shown that you keep suppressing IK1 below a threshold, you turn a ventricular myocyte into a pacemaker. So it's a very good candidate for that. If you don't have IK1, you have a pacemaker instead of a ventricular cell. But there are other mechanisms through calcium, a leaky ryanodine receptor or sodium calcium exchanger could contribute to this. So we have started to, this unpublished data, we have started to probe these different mechanisms in the context of cell density to answer the question, do we get more mature cells with higher cell density within the tissue context? So the example here is showing the effect of cell density on affecting the spontaneous frequency of oscillations as we start dosing them with different barium, probing, trying to reveal how much IK1 do they have. And at the low density, which is, so single cells are somewhere here. This is rather low density for the 96 well plates. We barely see any consistent effect to different doses of barium. However, at higher density, we start getting more and more engagement, as you would expect in a dose-dependent manner, revealing or telling me that perhaps these cells do get IK1 if you plate them, if they're in the right environment. It's sort of a new idea that in order to study certain ion channels, you have to take into account the context of the cells. It's not really, when you look back in the literature, there is maybe one AJP paper by, I forgot the first author, who has considered that maybe cell density impacts ion channel properties, but it's not a common concept. One can potentially study it with these new tools. We probed also the calcium-dependent mechanisms for these oscillations using a blocker for for the sodium-calcium exchanger, and that's expected. So we expect the low density here to be more responsive. In other words, to be able to block more effectively these oscillations as we apply these blockers, rather than these guys that didn't have leaky ryanodine receptors or had them to a lesser degree. Again, these are very preliminary data indicating that the high density, low density cells might be different by nature. So one more thing, as we are observing how the cells react, these optogenetic tools can actually help us maybe alter the phenotype of the cells. So the result of low IK1 or other factors on the iPS-derived cardiomyocytes are that they tend to sit at higher, at more depolarized resting membrane potentials. So in addition to the channel rhodopsin, we have this hyperpolarizing channel, RHT, which is a proton pump. And when applying light, you can sort of drive the resting membrane potential to more hyperpolarized states, which we have done at the single cell level. So this cell, upon application of light, we can forcefully hyperpolarize this cell by light. You can imagine doing this on a multicellular level, and we are developing, so these undergraduates that are developing this Arduino-driven light stimulators to be able to control this in the incubator, to either pace cells or maybe try to hyperpolarize them continuously and see if the voltage is a driver of maturity independent of their original ion channel expression. So collecting such enormous amount of data from millions and millions of cells, human cells, that can be made patient-specific, sort of opens the possibility to start thinking about constraining mathematical models in possibly a better way. So the general scheme of mathematical model development is we start with some generic cell model structure that the early developers came up with, and many of these fundamental structures were developed here. Then you do some parameter adjustment until you're happy with the prototype cell model, and then there are some experimental selected experiments that you use to constrain the model and use further tweaking until you call it a validated generic cell model of a condition. In the last 5 to 10 years, there has been emerging effort to do population EP modeling. That is, instead of a single generic cell model, develop these massive populations of cell models where you leverage the high throughput of computation. So computation is cheap. We can generate thousands of models, and then again loose constraints, some experimental data to further limit what out of this big population of models, what makes sense to call it a validated population of models. So the idea is that with this all-optical high-throughput electrophysiology that I have been talking about, one can get a step further and now perhaps have the high-throughput means, high-content measurements, experimental measurements, to get much more specific in the constraints at this stage, and to even derive personalized condition models that are fully based on measurements done on this patient's cells. So if I want a ventricular model for me, take blood, go through the reprogramming process, have my own myocytes, and ideally this model will be my model, reflecting what my genotype is and hopefully capturing me. One more thing you can do with that much experimental information that hasn't been available before, because you can throw all kinds of drugs onto this to reveal the ion channels, is to start thinking about identification of key molecular mechanisms. So in other words, kind of solving the inverse problem. I have all this data, I have some generic model structure in this population. Perhaps I can actually identify what the key parameters were if I probe the system rigorously enough. So it's a new tool that can improve computational modeling. So these are the four topics that, the 4 points that I tried to make, that this can be a new life simulator for cardiac electrophysiology. Can be, the translational potential is in the high throughput for preclinical screening and drug development and disease modeling. Can help with characterization and possibly maturation of iPS-derived cardiomyocytes, and can help with model building as well. We haven't done anything on the model building front of things, but the potential is there. And these people that are either currently involved or collaborators, Gil, Natalia, and Pat, and this is the financial support. Really, Christina and Alex were the main people involved in the projects I presented today, but there are other students that have been involved, and that's what I have. Thank you.
Thank you very much. Questions and comments?
Great talk. The cell density, do you think it's a gap junction issue, or do you think that it's actually modulating the channels directly?
I do not think it's a gap. So in order to dissect the cell density problem, we're thinking the following way. To perturb gap junctional coupling by whatever tools are available and see how things change that way. We haven't done that experiment yet. So in terms of gap junctions, these iPS-derived cardiomyocytes, I am yet to see a convincing staining for connexin 43, for example. They do support conduction velocities in vitro on the order of 12 to 15 centimeters per second, which is lower than, let's say, neonatal rat myocytes. So it's hard to say what role connexins play in these particular cells. I think is most likely release of molecules and chemical talk than the gap junctions themselves, but I wouldn't exclude the gap junctions because we haven't really done that.
If you plate myocytes at a low density, they start to die. They need something, like you said, that's created in the media. Maybe you could try and...
Yeah, one can go further and supplement the media from the dense, and yeah, you can expand this experiment to infinity. But the bottom line is whether it's certain concentration of molecules in the medium or some exchange of signals between the cells that they need. We haven't dissected that at all. But they're different. They're electrophysiologically different when they're plated dense.
And is there an upregulation of either the transcript or the protein, the KIR 2.1 or 2.2 in those cells?
So again, we haven't had the chance to do molecular biology properly. We wanted to focus on the IK1, but even that we haven't completed yet. So I'm sure many things changed, but we haven't really.
So arrhythmogenic responses of the heart depend a lot on, and cells depend a lot on regulatory pathways and all kinds of things that are not ion channels. And so, for example, take IKS block with or without beta-adrenergic stimulation is a totally different thing. And the question is how, when you assess drug toxicity, arrhythmias will develop in the presence of beta adrenergic stimulation, for example, in the heart, this can be taken into account in mathematical models also that have representation of those processes. How are you going to take it into account in these preparations that
Yeah.
you...
Right. So our thinking is the high throughput gives you a lot of power. So the high throughput means that I can have samples on which I can alter the hormonal composition. I can probably put attempt adrenergic stimulation, or I can do many perturbations to try to assemble a picture.
But
Because of the
for
power.
example, if you want to do adrenergic stimulation, how, you would add isopropernol to this preparation, or how? Because then you know, the isopropernol could be directly interacting with the drug that you're testing.
Yeah, I agree with you. We have done a neuronal cardiac culture where we selectively transformed either the myocytes or the neurons to be light sensitive and to be driving the system. So again, in this high throughput format to be probing. There are no reliable iPS-derived neurons that match the sympathetic or parasympathetic neurons in the heart. The ones that we used were rat-extracted primary stellate ganglion neurons that we co-cultured. So I don't really have a good solution how to capture all the complexity, but the fact that you can vary many conditions, it's helpful. If you come up with a solid idea, it instantly can be added to the experimental design and it wouldn't take much time to have that parameter tested.
I think we have to be careful, right? I mean, you want to be as simple as possible, but not too simplistic.
Correct.
And then the question is to be careful not to extrapolate too much into what is going to happen when the cells are in the heart. And so it's an important stage to maybe rule out drugs and so on, but then I don't see it replacing the higher level drug screening that is still, in my mind, still necessary, because the complexity, it's very difficult to take into account all the complexities.
Many drugs act on the vasculature, actually, and through altering the blood pressure.
Or heterogeneities and
systemic
iron
effects.
expression. So
Totally agree. Yes. Perhaps the diagram where we completely remove so the goal is to eliminate the animal testing part as much as possible, and to narrow
you
down.
cannot really replace it. But
To narrow down this funnel to not ever deal with compounds that basically are obviously pro-arrhythmic, which will save money and so on. But I agree, there are aspects that are not captured by this system for sure. Yes.
I was going to ask you about Titan. Do you know what isoform is expressed in these cells?
I do not know. I actually was looking, someone was looking into that. A woman in Germany, I want to say. But I don't know anything about that.
Yeah, I have a question on this illumination of the light in terms of intensity, in terms of the modulation frequency. Are those more patient variations, or is it quite homogeneous? Like you just use one type of wavelength modulation intensity?
Yeah, so we have done quite a bit of testing with, for example, to exclude generation of heat. So we definitely operate at light levels that are very low, under 0.5 milliwatts per millimeter square. And in terms of the actual modulation, for simple pacing purposes, we want the pulse to be as short as possible. We work with 5 milliseconds often, maximum 10 milliseconds to drive the cells, and it doesn't really matter much what kind of experimental model we use. As long as it's sensitized to the light, it's very straightforward pulses of light that you can drive them with. If we are talking about the first part of the talk, where we actually project a movie and we are controlling the waves, it's much more complicated. There we have the ability to create a gradient of light, which translates into gradient of inward current injected into the cells and different polarization of the membrane. So it's much more complicated story. You have to know how the channel works on the cells in order to predict what light pattern will produce what. So that's more complicated. But for the drug screening or these other tests, simple pacing by relatively short pulses of light with relatively low intensity seems to give a consistent response.
Okay, any other questions or comments to Emilia? If not, then thank you very much again.